appreciate it.
Thanks, Nikki.
Hey, good evening, everybody. Thank you, Nikki. And we are climbing in participants. I just want to give a thank you to everybody involved with this group right now because it is incredible. I'm a member of the National Association of Interpretation, and I've been involved with like some trivia nights where they do this kind of Zoom thing. And this is a national group, and there's like 8 or 9 people that usually join these things. And I'm like, we have this small group here and we have such great attendance. So it really means a lot to our presenters like Brad to have, have y'all returning time and time, month after month to hear these presenters because they do put a lot into, sharing their time and expertise with us. So I'm really thankful for all of you. And I'm feel very privileged to introduce Brad Jaynes. I've been fortunate enough to know Brad for many years now and lucky enough to learn so much from him and looking forward to the continuation of that. Brad is the critical area line project manager for the Bureau of Coastal Management, which is a division of the South Carolina Department of Environmental Services. Did I
Wow,
do
that
that?
was amazing.
I think I got it. Incredible.
We can put some more
Um,
words in there if you want.
there's a lot. There's a lot. But job title aside, he does important work with that. With that work, and many of you know Brad because several of you were lucky enough to have him teach fungi to master naturalists while you were either in class or maybe you've attended an advanced training. He's cataloged over 220 fungi species and other things at CAW using iNaturalist and other platforms. But I'm very thankful to introduce him to you tonight. And Brad, appreciate you taking the time to teach us more. We're always willing to listen. Thank you.
Of course. And I don't want to miss something if there's somebody else that wants to contribute something at the beginning here. And if not, three, two, one, then I'm just going to take it from here and let you know, number one, thank you for being here. I enjoy educating more than I enjoy most of what I do. It's always fun to go out and get completely away from people and just look at things that are not people, but I don't have to do that on a computer. I've just got a bunch of fungi in front of me. I would like to refrain from getting into any kind of slides or anything like that. I've got a lot of things that we were lucky enough to find yesterday when we did the Master Naturalist group at Kawkaw. It was a very dry week or two that we've had, and I did not think we were going to find much. That's a weird looking mushroom, Christina. But we did find quite a bit. And it does not matter if it has been dry for a while. If there is decent humidity and you've had a rain or two in the recent past, you may still find some things out there. And of course, what I mean by things are macro fungi, the things that are large and showy and colorful and easy to see. There are plenty of other things if you are really interested that involve things like crusts and films and parchments and resupinate bracket fungi and whatnot. If you really have the urge, there are lots of things, no matter the weather. They're just a little more difficult to identify, of course. And I say a little more difficult. I mean, you probably are going to invest a good bit of money in a really good microscope so that you can look at spores. It's not the kind of thing you can see with a magnifying glass or even a decent microscope. You need a pretty powerful scope to do that. I don't have that. And just to be clear, I am not a mycologist. I am not a research scientist. I am a hobby mycologist, for lack of a better term. They've always been interesting to me, but I also fell into this role working for the county park system because it was just a cool group of things and nobody was really attending it. And come to find out, it's basically the glue and the building block for everything else, to the exclusion of the bacteria and the other microorganisms that we don't usually see with our eyes. This is a huge group of things. We're talking about millions of species, probably tens of thousands just here in the Southeast. And we talked about it yesterday, the fact that there may be 800 or so bird species in North America. This is a much, much bigger group, of course. And if you find that you're interested in a particular group, that's great because it needs it. We need that help. Uh, Identification of species is what I'm going to focus on tonight because we found some interesting things yesterday. I've got some older, drier things that we can highlight. Hopefully, the camera on this computer is good enough for you to see some detail. But identification is a good first step, but it's also really important to me that you consider what these things do on the planet. And these might be things that directly benefit us. We might eat them, we might use them for medicine. These things might infect plants that we try to grow for food. They might directly affect us in those ways, but they might also very much indirectly affect us. So I don't want to get away from the fact that these are all ecologically very important organisms. They're not uh, things we should ignore because they don't have some immediate benefit for us. I'm not a forager. I'm not somebody who harvests mushrooms on a regular basis. I don't grow them in my garage. In fact, I'm coming to you from the Bicycle Museum, which is, I guess, a layperson's term for where I'm sitting. I'm in my garage with about 30 bikes. Uh, Bicycles are important to me as well. So you can ask me questions about that, but let's do that some other time. I'm going to focus on the mushrooms this evening. And I say mushrooms, I'm talking about fruiting bodies. I'm talking about the apple, the grape, the tomato, things that are the reproductive structure that these organisms form. These are only ephemeral. They're around for a little while and they're gone, except in the cases of some of the woody polypores, the brackets and shelves that you see out there that look like they're also made of wood. A lot of those are perennial and they'll continue to add growth rings year after year to those fruiting bodies. But a lot of what you're going to see this evening are things that might pop up for a week and then you go back out and they're gone. They're not gone perpetually, of course. They've come up and released spores into the atmosphere that have either traveled locally or in some cases across the continent or in very rare cases across oceans to other continents. This can happen because these spores are incredibly small things. We're talking about things that are the size of what was it, dust mite feces, I think, that we had on the slide in the classroom yesterday. 10 microns, so things you can't see, as I said, without a really good microscope. Without further ado, I'd like to get into some of the things that we saw when we went out yesterday, but I would encourage you in the future, for example, this weekend on Saturday, if you're coming out, if you have something that's growing in your yard or you have something that you have a photograph of, feel free to share that. I think in future engagements like this, we might just do show and tell kind of moment or two, just to make sure that we're covering some things that maybe are where you are, but we're not seeing because we're out at Caw. A lot of things pop up in people's yards, along the sidewalks, sometimes in your homes, unfortunately. That does happen. But I'd like to know where you'd like to start. What's a group that you are interested in or that has perplexed you, or maybe somebody just volunteers something you've seen lately that you would like to sort of feature as our initial specimen here. Maybe I've got it. Maybe I don't.
Um, we were out at Kiowa doing a bio blitz last week, and I saw
Right.
what looked like a tiny little, comparatively tiny little lion's mane, and it was in clusters. So it had the
Right.
same shagginess of a lion's mane, much smaller than anything I'd ever seen, and much more. I mean, it had the feel, first glance, as a turkey tails kind of looking. I mean, it was just a bunch of little ones. Very weird.
Shaggy like toothy.
Hmm.
Okay. And we do sometimes see that you might have fruiting bodies kind of merge when they're really young like that. There is another species in that genus that we get occasionally that turns into almost like an antler of spines like that, uh, or a branch of spines, as opposed to one big ball of spines like a lion's mane would. I have a lion's mane, but I will say it's one that is quite gone in terms of its aged nature. If I can find that.
A couple of pictures too. I'll bring Saturday. I'm working Saturday, but I'll be able to see you.
There's a little bit of mold coming in on this, which is, of course, another fungus that is decomposing this lion's mane. But I'm trying to show you the side that does not have mold on it, and I'm hoping that you can see some of the detail in this image. I think this camera is fairly decent, and I'm hoping that you can catch some of what I see. I haven't maximized my screen. Maybe I can do that um, so that I can see whether it's got decent detail there or not. Well, I'm not going to worry too much about that
It
in
looks
a
good.
week's time. Uh, just let me know if you do see any spines or teeth to that thing. They're kind of laying down. It's sort of matted at this stage, but this form is sort of what you'll see with lion's mane. It's almost kind of an upside down bullet shape that's really broad. It's usually pure white when you see these and they're fresh. There is one at least, I think two. Christina saw another one out at Caw Caw right now. This is one that more recently has been shown to have some medicinal efficacy. So it's not something that is an old grandfather's tale, so to speak. It's something that has been shown in clinical trials to improve, if I'm not mistaken, cognition or memory and or memory in mice. I'm not a mouse, neither are you. Uh, I would say that there is still more research to be done about that to conclude that this might be something that would be beneficial for us. And I only make that distinction because this is a group that contains quite a few noted medicinal species of organisms. A lot of these things have been studied, if you will, in other parts of the world for a long time, and some of them do seem to have some real effectiveness, but very few of them have gone on through clinical trials, and some of them have gone through none. Uh, Most likely, a lot of them that are noted as being sort of panacea type species are probably immune uh, boosters, if you will, things that might help somebody at least keeps an illness at bay or at least speed a recovery for someone with a particular illness. But I don't mean to depress you about that topic. There certainly is plenty of promise in that world. You just need to look at things like penicillin and cyclosporin and other drugs that are used regularly in the medical industry now that have saved all sorts of lives. So we know that this is an unexplored or underexplored topic, but lion's mane is one of those that you will see in places like Caw, bottomland hardwood swamps, on oaks usually. I typically see that somewhere around head height or higher, although it does occur lower than that. I had it on a tree that had fallen over next to a drainage ditch at Caw yesterday. So just because it's usually in a certain place doesn't mean it's always going to be there. But that's helpful to note that sort of thing. It's helpful to note all kinds of details if you're trying to identify something, especially to a species. I'm going to try to stick mostly with just a genus this evening. This is also a really plastic group. There is known to be horizontal gene transfer in this group. There's known to be some real variability within a species. So I don't want to get too focused on that and say that this is absolutely this species because I'm not convinced that I know that about almost any of these this evening. Maybe a handful, but I'll stick with the genus for the most part. If you hear me uh, say a species name, then you can smack my hand with a ruler the next time you see me. How about that? The other thing I want to mention before we get started here is pronunciation of these names is not that important. I say it's not that important because the more important thing is you know how it's spelled so that you can look up a reference in a book or so that you can communicate this to a naturalist or mushroom observer or some other open platform like that. Uh, a lot of times these things are already in those bases and you won't necessarily need to spell them, but a lot of times people incorrectly spell these when they're putting their identifications on those platforms. So if you fine that it is easier to call that an amanita because that's how you spell it. That is fine. Because someone calls it an amanita does not matter. No one's going to give you a hard time about that. And if they do, then don't hang out with them and go find another person to get in the woods with, is my advice. So, lion's mane is a fairly common one. It is an edible species. I'm not a big fan. I like more of the umami flavor that you get from some of the other things that we have locally. Doesn't mean that it's going to be a bad taste to you. Everybody has a different sense of smell, different sense of taste. So, it's one that is pretty distinct. There aren't a lot of things that look like it. So that is not a bad one to focus on if you're thinking that you might go that direction with this interest. But I would certainly use a field guide and try to use a key if you're trying to identify something, or just bring somebody who knows what they're doing. That would be helpful. I don't mean me. I'm not for hire. Again, this is not my goal with this. I do sample a lot of these things so I can talk intelligently about what these tastes are and the textures are. There are plenty out there that are listed as edible that taste like garbage to me, others that feel like shoe leather when I chew on them. Uh, just because it's edible doesn't mean it's good. So keep that in mind. I'm going to start with one that is fairly easy to identify for some of you, maybe. And that's this one. And I'm going to start with kind of a larger view of this. And then I'm maybe going to flip that and show you the underside of that cap. This one has a really long stalk or stem or stipe to it. Again, it's good to know these terms that are references to the structures, but don't get focused too much on that. If you call it a stem or a stalk, that's okay. We do have a stem or a stalk that looks like it has quite a bit of texture to it. I'm hoping that you can see that in that image. And we do have a cap that tends to have a lot of texture to it. You can see it almost has like scales to it. In fact, the second portion of this name is Lepidius, which is a reference to that scurfy, scaly cap and stalk. Uh, the underside does have gills to it, but you will find this one listed in a polypore reference book because it's more closely related to those. Just because something has gills does not mean it's not related to that shelf fungus that you just saw. And just because something has pores does not mean it's related to another thing that has pores. Can somebody give me an example of something that has pores that might not be related to, say, sulfur shelf or chicken of the woods or turkey tail? There's a whole other group out there with pores on the undersides of caps. Anybody know what group I'm talking about?
Bullet.
What is it? That was boletes, I think
I
is what you
thought
said.
I was going to guess boletes as well.
Yeah, so the
Yeah.
boletes, the slippery caps, slippery
Hmm.
jacks, I'm sorry, uh, the Tylopolis, the Bolatelluses. There's quite a big group of capped mushrooms that will have pores when you look underneath them. Again, that doesn't mean that there's a taxonomic relationship between that and something else that has pores. That's not a real important aspect unless you're using particular types of keys. We're going to use one in a moment here with one of these that focuses on whether it's a polypore or not. And many pores is a good description for a bolete. It's a good description for a slippery jack. Those do have many pores, but they are way different in their form. So that might lead you down the wrong road. So just remember that we're not talking about things uh, that have the same form, so they're related. That used to be the case with a lot of living things that we would describe that external morphology, that external form, and say, well, that looks a lot like this other thing, so it must be a relative. Sometimes that's the case with this group, others it's not. Uh, this one was growing on a huge pine tree off the driveway into Caw, and it actually had acquired some of the smell, I think, from the resin that it was growing off. Uh, this one typically will grow on conifer wood, including creosoted wood, which is frequently used underneath steel rails for trains. Does anybody know this one? Let's imagine that this consumed lots of those creosoted timbers under those steel rails and compromised the functionality of that train track. What would that result in?
All right, I'm going to call it a train wreck.
It's a train wrecker. One of the common names for this one. Neolentinus or Neolentinus is the genus on this. Um, and it's pretty characteristic. There aren't a lot of things that look like this, especially with that really long stem that this one has. That's pretty unique. I don't see a lot of typical quote mushrooms with really long stems like this. There aren't a, it's not unique, but there aren't a lot that have that. Train wreckers are fairly common, but they're kind of seasonal, and we did not see that one yesterday. That's a dried specimen that I showed you. I have another one here that is also described in a polypore book, but if you look underneath, I'm hoping that's clear. Thanks for nodding, Christine. I appreciate that. Or you can give me a thumbs up if you want. The gills that you see there might lead you to think this would not be found in a polypore book, but again, it's got a taxonomic relationship, a DNA relationship to the others that do have pores on the underside. And this one is pretty cute. You might notice that it's growing in a group, and that would be a good thing to notice. That train wrecker was growing by itself, a single fruiting body by itself. This is a group, a gregarious group or a confluent group. And the caps of these things have some real texture to them. I'm hoping that shows up. It almost looks hairy or fuzzy or furry. And Panis neostrigosus is the name on this one. This one is one of my favorite common names because I always enjoy saying hairy panis and being misunderstood when I say that. Uh, this one is beautiful when it's fresh. Right now, it's totally brown. That happens a lot. And it doesn't just happen after these things get dried. You will see what we call a hygrophanous response on a lot of mushrooms where they look very brightly colored when they're fresh, they dry out, and they totally change color. This one, when it's growing, has almost a violet color, a beautiful kind of purple hue to it that is not at all evident in this dried specimen. But what else might you notice about this one that you did not see attached to that train wrecker, even though we know that's growing out of creosoted wood? This one is attached to a piece of bark. This is a decomposer. This is a saprotroph. This is something that's going out and gathering easily available food, just like that train wrecker is. That's an important thing to notice. If you are going to collect something, I would advise you to bring a garden trowel or something like that with you and try to get underneath the base of that mushroom because a lot of times that's going to be very important for your identification. This is another thing where another aspect that I differ with on the foraging thing. You got people that might go out and just cut that stem in half just to bring that cap back to consume it. Well, without that base, you've lost something that might help you identify that fungus. So it's always a good rule of thumb just to dig that thing up, even if you do chop that top off to take it home. It's not a bad idea to get that base and see whether you have any kind of evidence there of any kind of a universal veil. That would be a sheet that basically covers that whole organism as it's developing, that whole fruiting body while it's developing. We might see one of those in a moment with another one that I have here. But I want to jump through some of these so that I don't keep you all night. I want to start with this guy right here. And this one has a quite different form from the other ones that we've been seeing. We've got what might be referred to as a funnel form. It almost looks like the gills are a little bit decurrent. They go down the stalk a little bit. They do, but not quite like something, say, a chanterelle or something along those lines. I will give you a clue and tell you that when we collected a few of these yesterday, we did scratch the surface of those gills right around the base of that stalk, and we saw a latex response. We saw some droplets form that look almost like Elmer's glue. So, what kind of mushroom is this? Anybody?
I'm gonna let them guess first.
The milky something, isn't it?
You got it.
Yeah, you got it.
It's a milk
Oh,
cap or milky cap. Ironically, of course, it's very rare to see that milk on the cap of the mushroom. It's usually under the cap on the gills where you see that response. And that's probably something that helps distribute spores to other places by getting stuck to the legs of beetles and flies and other insects that get underneath there. Springtails, we saw one of those yesterday on the underside of one of these. Uh, it also might be something that helps prevent those things from eating the mushroom. If they do taste bad, then those insects are much less likely to eat them. Um, it might be something that chemically is aversive for those insects. The milk cap group, I found yesterday that the easiest way to describe this group is that it's a clean mushroom. You don't see any kind of remnant universal veil at the base of that stalk where maybe there was a almost like a ripped bag at the base. There's no partial veil on the stalk that would have been covering the gills while it was developing, because these things basically protect the most important part of their bodies until those spores are ready to drop. So once this thing has ripped that partial veil open and sometimes leaving a little bit of it on the stalk, now these gills have spores that are probably close to maturity, probably close to being ready to go out into the world on their own. So milkcap, a very common group. We have quite a few of them in our area. They have a clean aspect to them. A lot of them have what we might call concentric rings in the cap. A lot of them are referred to as zonate. We have one called the sand milk cap, the Lactarius samicola. It's got really prominent rings of yellow and orange and almost brown to it. It looks like a little bullseye when you see it fresh. But again, that hygrophonous aspect, you might lose that color over time. A lot of these things, as they get older and drier, lose the prominence of that concentric ring. So don't think it's always going to be there. It's just helpful if you see that to help identify that as a lactarius. But if you see the concentric rings and you scratch those gills near the stalk and you see some latex droplets, whether they're white, whether they're blue or green or yellow, it looks like it's starting to bleed. That's probably a lactarius, especially if you don't have that partial veil and that universal veil at the base. Kind of a clean aspect. And I would say the same thing about these guys. This particular one is kind of small. I'm sorry I don't have a larger one, but this group is related to the lactarius group. And this one you've seen before in a red capped form, I'm sure. It's one of the most noticeable mushrooms in the forest. That Rusula emetica and sylvatica group, uh, lots of red caps and white stalks and white gills. The Rusula group has a lot of different species, and you are going to see these out there, but it's tough to identify these. A lot of these are not noted as edible or noted as causing gastrointestinal distress, so not a great group to focus on if your goal is to find food, uh, but easily identified again because of that clean aspect, kind of a neat aspect, I should say. Clean's a bad word to use because a lot of these things are growing up through leaf litter and soil and duff and sand, and some of them are going to come up looking like they've been tossed around in a compost bin. But that neatness is what I'm focused on here. This particular one had some granular sort of texture to the cap. I'm not sure that that's obvious in this one, but that's kind of different for Rusula. You don't see that a lot. And most Rusulas have what we would refer to as striate edges to the cap or radiate lines from the center. So instead of the concentric rings in groups around that cap, you might see that the edge of that looks like it's had a lot of lines scored on its edge away from the center of the cap. That's usually pretty noticeable in Rusulas. It's not in this one. And I think I'm looking at a Rusula compacta or something related to that group. I'm not going to make an ID on it. I just want to make sure that you understand that Rusulas are very similar to Lactarius. Brittle gills is a common name for these because if you do crack one open, it breaks in many places. It's not fibrous, it's not tough. No offense, mushroom, but It is a brittle flesh and a brittle cap that will easily break into lots of little pieces. You really see that when you start breaking the gills off. They flake off like bonito flakes or something. Um, Really common group, and just like Lactarius, these are mycorrhizal. These are not eating dead things that are laying around on the forest floor. They're tapping into the roots of all of our vascular plants for the most part and exchanging moisture and minerals in exchange for carbohydrates or sugars that those plants are able to make on their own. And I'm sorry, somebody had a question, but I'm not sure I saw what that was. So maybe someone else who is on the
Yep.
meeting can answer
I,
that.
yep,
I
I
can,
can read
oh,
it for sure. Oh,
Okay.
sorry, Christina, do you want to read?
Sure.
no, go
Okay,
ahead.
I can read it. So Mike asked an aside question, have you or any of the other members observed downy woodpeckers foraging on fungi from the bark of an oak tree? I examined the fungi and surrounding area and there were no insects present.
I'll bet there were insects in that fungus. These things are irresistible to insects, and it doesn't matter what species. That one we picked yesterday that was the elbow patch fungus, it's like a big knot of wood hanging off the side of the tree. It's practically the same consistency as a piece of soft wood. Um, and I don't mean soft wood that's rotten. I mean soft wood that's, you know, closer to something that's not an oak, closer to something that's like a pine or a spruce or something along those lines, or a juniper. Um, you can knock on it, and it sounds like you're knocking on a door. But that fruiting body I pulled off, Christina and some of you saw it, perhaps. I'm not sure who was there, but it was perforated and looked like a shotgun had been blasted at it. There were holes throughout that piece of, quote, wood that was really a fruiting body that was a mushroom. So I wouldn't be surprised that it was going after things like that that are inside of that fungus. And I partly say that because those are known to do a lot of foraging on like goldenrod galls for the flies that are inside of those galls, Cecidomyid flies. They do a lot of things like that. They're inventive food finders, and it's not just those goldenrod galls, but that's something that's been reported. I think that's in one of those books you have at Caw, Christina, that Field and Shore book or something similar to that, Forest and Thicket, something like that. Um, so on that note, if you do forage and you bring something home, I would advise you to at least give it a little salt bath dip or maybe an ice bath dip just to try to at least scare off some of the things that are on those fungi. If you're squeamish at all, you're gonna have some flies, you're gonna have some beetles. You can cook those things up. They're not gonna hurt you in any way, but if it's the kind of thing you don't want to see while you're cooking, it would be worth your time to wash them at least. I like doing an ice bath. Somebody else suggested a salt bath yesterday. I don't like to run them under the sink for 10 minutes or anything like that. Um, I like to preserve that thing in as natural a state as possible, and ice bath tends to do that because you can just dunk them and pull them out. I would not do that with this mushroom. Why would I not do that with this mushroom? This is a mushroom that has a cap with some granular wart-like things on its cap. It has a partial veil. This one's an old dried specimen. It's got a little bit of mold on it. That's not the reason that I'm talking about not eating it. There's also remnants of a universal veil at the base. They're not as evident here because this is one that doesn't turn into a sac-like form at the base, but it does have almost a leafy aspect when it's fresh. Um, but as it dries, it kind of adheres to the stem a little bit, kind of flattens out. But because this has the universal veil at the base, it's got a partial veil on the stalk that was covering the gills. It has some evidence of the universal veil still on the cap. And if this was fresh enough, you would see that it also has some striations at the edge of the cap. This is an Amanita. And those things I just mentioned are the things you need to focus on for this group. The other thing that you can still see, even though this one's degraded, is that it has pale gills. They're usually pure white gills. That white gill is a very telling sign. There are things like agaricus. Agaricus is the genus that contains button mushrooms and portobello mushrooms and things like that. So it looks a lot like it when it's growing up in a grassy lawn. Well, this one is mycorrhizal. It's growing with a living vascular plant. Those agaricus are not doing that, but they might grow similar places to this one. The white gills, however, would be very odd to see on something like agaricus because those at the palest are like a dark pink. They turn into a chocolate brown over time, though, and that's a really easy thing to see if you just rip off one portion of that cap. But Amanita, I think, responsible for 95% of lethal consumption with mushrooms is that genus. I think it's 50% of people who get sick from eating things. And a lot of that is probably people mistaking the Amanita group for agaricus because there are meadow mushrooms and others that look similar to at a cursory glance. But to look a little more carefully, you would see these are white gills when they're fresh and they mature into a white color as well. It's not like they turn color over time. This one kind of has because it's a little bit old. But the agaricus would never have those white gills. I shouldn't say never. Someone could spray paint it. Another one that we found yesterday that is in that same genus does not have a partial veil evident in this specimen. We saw them yesterday, but they were really fragile and they fell off. Again, something to note is take a look at that thing in context. Take a photograph of that before you touch it so that you do capture that partial veil in your photograph. And again, don't just slice the stem off. Try to dig under the base of that thing. And Christina, you saw me do this yesterday. Before you even get that trowel in there, you might want to at least peel back some of the leaf litter at the base just to see if you notice the remnants of those of that universal veil because this one had it, but it wasn't real prominent. This is one of those that the veil kind of sticks to as it matures. But this one was also full of like a powdery white substance, and it was all over the cap, all over the stem. It almost looked like somebody had sprinkled powdered sugar everywhere. Most of that has been shaken off since then, but we do see some sort of scaling or warty aspect to the cap, and those are again remnants from that universal veil. So this would, in a fresh form, have a partial veil, have remnants of a universal veil on the top, some at the base as well. I don't remember seeing the striations on this one, on the edge of the cap, but that's the least important of those three things I just mentioned. The remnants up here, the remnant down here, the remnant here, and the fact that this is pure white underneath when you look at the gills. I'm hoping that shows up. There's some soil in there, so it may not look quite white anymore, but agaricus would not have the white gills. So Amanita, very big group. None of them noted as good edibles. There are some that are listed as edible. And part of that is because there's a history of people eating all kinds of mushrooms. And in some cases, people are just using references from the 1800s or earlier. I'm not sure that I want to use that sort of information anymore in 2025. We've had a long time since then to figure out whether people are getting sick from these things. But it's also really important for you to understand that we all have different constitutions. We've all got different chemistries. We all maybe consume alcohol with our mushrooms or do not consume alcohol with our mushrooms. There are other things that can contribute to the toxicity of these. So keep that in mind. I know I keep coming back to edible things, but that's largely because I know that all of you are going to run out and grab a mushroom tomorrow and try to eat it. I'm joking. You really want to make sure you're paying attention to properly identifying these things. It's not as if most of them are going to kill you. That is not the case. There aren't lots and lots of them that will kill you. There are plenty that will make you sick, and there's a handful that are choice edibles. Plenty that are edible, but why would you? Because you'd have to collect baskets of them to get enough to have a meal, and there's really no point with some of those. I'm going to try to cover the keyboard of my computer here. I feel like I'm getting a bunch of stuff on my keyboard. What do you see at the base of this? Can anybody see that? Looks like spider web almost. That's not spider web.
Oh.
That's the body of this fungus. That's the mycelium of the fungus. It's a, a group of hyphae, which are the growing tips, the strands, individual strands of the body. Sometimes those look like a spider web. Sometimes those turn into the thing that you see at the top. This is all mycelium. These things are all formed by the same tissue. It's just that they're inflated with water. You occasionally wind up with some change in the cellular walls on these things, or membranes, I should say. But anybody know this one? Let you know the group. If I tap it, you can
It's
probably
a
see
puffball.
some of
Is it
the.
really a puffball?
Can you see the spores coming out of that?
Yep.
Yeah.
Just a tiny little cloud of brown spores, millions of spores, even in an individual fruiting body. So we're not talking about 100 or a few hundred or tens of thousands. This is an enormous amount of spores in here. This particular puffball is Apioperdon. This is one that, gosh, I can't remember there's a common name. It's a pear-shaped puffball is the name for this one, I think. This one's everywhere at Kaw right now. A lot of it on dead wood, decomposing. Raindrops hitting these things and hopefully making that puff happen so that those spores can get new places. Most of the things you're going to see are airborne spores. There are plenty of others that use things like latex or maybe the goo that you see on stinkhorns to help physically carry those spores to other places with the help of beetles and flies and things. But this is one of those that's definitely airborne and definitely in my lungs now because I'm standing or sitting right here next to it. I'm not concerned about that. There are some fungi that have been shown to be able to exist within our bodies, but most fungi are not able to exist at our internal body temperature. It's a little too warm for them, which is a good thing, but there are some. So I would say when you are checking for an odor that is listed in your field guide, take it easy. Don't take a deep breath. Just a little sniff, if you will. And for everyone's response, I show you this. Anybody?
That's our chanterelle.
Those are chanterelles. And again, you'll hear people pronounce it differently. That does not matter. I'm not pulling out my ruler. This is probably the Cantharellus confluens, and I say that only because you have a confluent group of fruiting bodies. You don't have an individual fruiting body and then another one a few inches away and then another one a few inches away. This is a cluster of that species. That's a good guess at what this one is, but the Cantharellus genus is known to have lots of edible species, so I wouldn't be too concerned if that's your goal. It's fun though to try to figure these out two species if you can, and sometimes it's just what is the habit? Is it growing singly or is it growing in a group like this? Um, the key thing with these, of course, and you've probably heard this before, but they do not have, quote, true gills. It's not as if these gills do not have spores on them. They do the same thing that, quote, true gills do, but they do not have that pages in a book aspect to them. They don't look like leaflets all stacked together in a row. Um, you don't have a lamellae here, you have grooves, basically. Um, So chanterelles, mycorrhizal, just like the Lactarius and the Russula that we saw, the Amanita that we saw. Do you see a pattern here? All of our vascular plants are being helped by all of these things and thousands of other species. These are making those things healthier and making themselves healthier. It's not as if it's an even trade all the time. I'm sure there are times when these are getting more benefit and the plants are getting more benefit at other times. But it's a good sign that you're seeing lots of fruiting bodies. It probably means that those trees or those vascular plants are doing pretty well. Or they're on their last gasp. So this is a cool one. We don't see this one a lot, and that's partly because it's pretty quick to fruit. Um, I noticed this one because it has kind of a lumpy cap. It looks like It's got lots of raised bumps to it. It doesn't have that sort of smooth dome to the top. I'm hoping you see that. Almost looks like a sheet of uh, the top of the queso pot when you've turned the heat down. That's what it kind of reminds me of. But the underside is very different than everything else that we've looked at. And I'm hoping that you can see that this is not gills that I'm showing you. Oh, it says mute, but I did not mute. Sorry, it's asking me if I'd like to do that. This is a toothy underside, and this is one that's referred to as the hedgehog mushroom. Can you see any of those little tiny teeth on there?
Yep.
It's really nice to have a hand lens with you when you're out in the field because sometimes this is not immediately evident. If your eyes are kind of bad, and mine are, that's why I've got the glasses on, and I picked this up and looked at this with just my eyes, I'd probably look and think that I was looking at pores from a bit of a distance. But when you get a little closer, you can see that it's actually lots of little stalactites, basically. Um, so these are tiny little projections that again are doing the same thing that gills do. They're bearing those spores on those projections. It's just another way to increase surface area and increase attachment space for those spores. So you're no less of a mushroom. This is the hedgehog mushroom. This is Hydnum. Uh, we do have Hydnum subolympicum in our area. That's probably what this one is, but I'm again not going to stake my fortune by that. Uh, we've got a really nice one next because we have a spore color right in front of us on the mushroom itself. And I'm hoping that you'll be able to see that color on this one. This one is a great example of this genus, and this is a genus. Let me show you that side. That's the better side. This is a genus that is named after a structure that's on the stalk. So we know that's a partial veil, right? It has a sort of a tissue around the middle of the stalk there. I'm again hoping that you can see this.
Yep.
It's kind of a webby partial veil, though. It looks more like spider silk or something. It doesn't look like a sheet or a skirt. It looks more like cottony masses of threads. We refer to that as a cortina. And even when this is developing, it does not have a sheet like an Amanita has or like some of these others have. It has just a thready little mass around that thing. And the nice part is it's really good at catching spores. And you can probably see the color of those spores, I'm hoping, um, is orange. It's kind of a rust color. And that Cortina immediately tells me that this is a Cortinarius. That genus is something that is unique in terms of that structure I'm talking about. That Cortina is not found on anything else that I know of. Uh, Unfortunately, this is a genus that includes over 2,000 species just in this single genus. That's worldwide, that's not in our area, but identifying this two species would be a little more difficult than just saying it's got a cortina, it's got this color of cap, because this cap looks kind of pale to me now. This could have been one that was kind of a violet color originally and has just changed color over time. And if you can see, I'm not sure if you can, there's always a bit of a sheen to the Cordonarius group that looks kind of like silk, actual silk. It reflects a lot of light and almost has kind of a shine to it, um, that I think is really easy to see in person. I'm not sure if it's coming out on the camera, but that does not just extend to the cap surface. You do see that on the stalk as well. I got another one from the same genus here, and you can see that kind of silvery sheen, I hope, on this stalk that I'm holding up that's kind of split, and it looks kind of fibrous. That fibril or fibrous aspect to the Cordonarius is also pretty diagnostic. I wouldn't say it's unique to this group, but the Cortina, the silvery sheen with the fiber-like look, the rusty colored spores, that's important, all of those things. The cap color, not so important. In fact, that's probably the least reliable thing that you could focus on. It's a good thing to note what you see, but it's not something that's going to carry you to an identification. Um, I've got a handful of others, I think. I'm going to go a little quicker on these. I've got one of the sclerodermas here. This is one of the, uh, some people might call them puffballs. We had Christina and I found a common name for this one that I enjoyed. It was potato earthball. It does kind of look like a little potato when it is intact. Um, but you do see some mycelium here on the base, almost a little bit of a stalk there too. And this also has kind of a casing to it, if you will. I'm not sure if you see the thin tissue around the spore mass that's in the center there, but those things are different from what you see on this rhizopogon, which I think also could be aptly called an earth potato or a potato earth ball. It looks a lot like the same kind of thing, or looks a little bit like I'm holding a piece of feces in my hand. This is rhizopogon, which is basically translated to, uh, well, rhizome, right? You know that that's root. We've got rhizome with the same root, and we've got pogon, which is this guy right here. So we've got root beard because when this is developing, it typically has one little tiny tuft of mycelial threads that attach it to what it's eating. But it's real fine and really loose, and it very frequently will just rub off over time because the friction of this rolling around is enough to scrape those loose. But where you saw kind of a casing to this scleroderma, you don't really see that on this rhizopogon. This rhizopogon is more of a spore mass that looks like it goes all the way to the edge. And those things are important. These look like they're just floating around almost like a tumbleweed or something a lot of times. But I will note that I did have to pull the scleroderma up out of the ground, whereas the other one, the rhizopogon, was just kind of flopping around down there. Maybe a good idea to touch that with a stick before you pick it up with your bare hands. Um, we do have this guy, and this is a very dried specimen, but I'll turn it over because it typically does have some yellow and orange hue to it when it's growing. And just to give you a better perspective, it would be growing out of a log like this, of course.
Feedback. I asked for feedback a long time ago and got nothing. So, hello, people.
What's that?
Hey, Karen, you're not muted. Karen
Oh,
may
Is there a question?
No, I think Karen and Louise Sawyer aren't muted. They might want to mute themselves just to not have a I'm seeing Jane Ford too might not be muted.
Yeah, I'm
Just in
sorry.
case. And
I'm
that's
sorry
okay.
if somebody's texting a question or something. I'm not seeing any of that right now. I'm trying to stick to what I'm doing. So if you did have something, please speak up. I'm happy to address those questions. I am not trying to dominate anybody's time here. I'd like to help you learn. So just let me know if you have something. I'd be happy to engage.
You're great, Brad. What we do actually, and you just kind of touched on it a teeny bit, but Nora had a great question about, she said, when out and about and you want to look for fungi and mushrooms and you're a newbie, do you recommend wearing gloves for folks that are really new at this and might not, quite feel like,
I
is this a
I
piece of poop or is this a mushroom? I don't know.
I think that's fine. But more because you may be putting your hand where there might be a wasp or yellow jacket or, something else that you might not want to touch. It's not because the fungus is going to be a dermal irritant or anything. Some people do have more sensitive skin, and there is always the chance that you're touching something that's got chitin in it, and you might have a slight allergy to something like that. I'm not familiar with any records of anybody having any serious reactions to anything like that, but you know, encourage you to do what you feel safe doing. And you could make it just a light cotton glove or something like that. It doesn't need to be more plastic for the world. You can do something that's just a protective barrier.
That's great
Just the-
suggestion. You also like your use of the trowel too, or like a tool that can help is really, I think, a good suggestion too. Um,
yeah, trowel keeps you kind of hands free at least until you've got it up out of the ground, and then you know that you don't have a cicada killer hanging out underneath that thing or something. It's not a
that's great
not a pleasant
advice.
sting.
One other question from Jerry Vane. It's a good question about the purpose of the universal veil. Is it for protection? Is it
It is.
functional? What's the universal veil all about?
It is. It's just a little house that protects that fruiting body from some of the things that would be eating it before it was ready to emerge and produce mature spores. Because again, if it gets consumed at that stage, it all bets are off. It's not going to produce any spores that are viable. So it still needs more development. So it's basically just a protective casing. And the one I was showing you is one that a lot of you have seen. This is sulfur shelf latiparus. This one is Latiparus cincinnatus. It's got a white underside to it. A lot of people would just call this sulfur shelf because it looks yellowish when it's fresh. That does not mean that this is a single species group. We have, I think, 4 or 5 Latiparus in our area, in the southeast, I should say. But this one and sulfur shelf are pretty common. It's nice, though, if you look at your field guide, and I'll talk about one in just a moment that I recommend. This is one that typically is found near the bottoms of trees or out of stumps. This is not one that's typically high up. The sulfur shelf is usually higher up in the tree. So that something as simple as that can help you with separating these out. But that whitish underside compared to a yellow underside, it's a very different appearance when you see these fresh. So it'd be easy to make the call on that. Sulfur shelf is a polypore. It is decomposing, just like the turkey tail that I've got here. And we saw this one in the classroom yesterday. There are lots of turkey tails out there. This is just one of them. This is the versicolor. This is the one that has the, it almost looks like a turkey tail in reality. This is the most colorful one in terms of being the brightest, but by no means this is the only species. There are plenty out there in the woods. We've got at least 5, I think, that we see at places like Caw. But keying them out is the key on these. There is a roughened underside to these turkey tails, distinguishing them from the false turkey tails. It doesn't mean that it's any less of a mushroom if it's a false turkey tail. Those are both decomposing tons, literally tons and tons of wood every year, turning that stuff back into soil that other things can use. So keep that in mind. Um, we do have Ganoderma. This one is one of our Ganodermas. This one is also known as reishi by some people, but that's a common name that probably is more appropriate for a species from a different continent. The Ganoderma group in our area, we at least have a few that are pretty common. This one is the sessile form. You can even see that with the way that broke off of where I collected it. It does not have much of a stalk to it. There is a stalked version, Curtisii, that we get in our area. This one's Ganoderma sessile or sessile. Uh, these are both in that genus that is also used medicinally by a lot of the world, not as much by the United States or North America. But if you buy something like MycoShield or one of those immuno sprays that you can put in your mouth, it's probably got Ganoderma in it, in addition to chaga and all sorts of other things. Um, we do have one more polypore here that I also found today, Microporellus. We found one of these yesterday, but it was not the same species. This one is Microporellus obovatus, and I don't know if you can even see pores on this. Microporellus is a good name for this. It's like 6 to 8 pores per millimeter. So it's very, very tiny little pores that you see on the underside of this. But what I really like about this is that you can really see a stalk to it, which is not always the case with polypores. And you can really see zonation on these caps. I've got a log that I brought with more of these on it. And I think it makes it really easy to see the zonate cap with those, again, kind of like concentric rings going out from the center. That's important to note when you're trying to identify something. Might also note that that's growing on a dead piece of wood. And this is a piece of hardwood. That's also important, but not the end of the world if you miss it. Um, Keep in mind that you don't want to get too focused on whether it's a conifer or a hardwood. but a lot of times you can identify those even if they're really degraded. They've may have lost their bark, but there's usually a pretty different look to an old hardwood that's lost bark as opposed to a conifer. So I got one more kind of treat for you, and it's one that has this bulbous base that you see here. That is not the entire mushroom. I'm not trying to play tricks here. But I cut the top off of this because this is one that I knew, but it's one that gets mistaken for this group, the Cordonarius group, because you might notice that that's also got a bulbous base at the bottom. I've got my fingers in the way. Sorry, you can't see that other one. The stalk looks real similar. They have kind of a shiny aspect to both of them. Uh, we do have some Cordonarius that have a violet or a blue or indigo hue to them. And when this is fresh, guess what? It's also blue and violet. But look at it now. And this is probably just a day or two old, but it's been sitting out in all that wind that we've had. Our humidity and dew points have gone down. And so it's just been out there drying out. Uh, but this is an edible species, and it's one that not a lot of people have tried. I don't know if you can see the violet hue to the underside. It was kind of weird earlier where if I hit just the right angle with light, I was catching some of that violet hue, but at other angles, I couldn't see any of it. They basically looked like little tan mushrooms to me today when I initially saw them. But this is in that same group that includes tricholoma and some others that are very similar in form. And again, I don't see a cortina on this like I would on a cordonarius. And cordonariuses are generally going to make you sick. I think all of them, for the most part, that we have in our area are not worth trying. But this one is noted as a choice edible, usually kind of purple, usually kind of violet. That's lost now, but this is one that was just decomposing. It was not mycorrhizal. Uh, you, how do you tell that? You look at your field guide. It's pretty tough to figure out whether that mycelium is connected to the roots of a vascular plant. That's a difficult task, but I know what this is. I looked it up to confirm the ID characters. I'm not sure if anybody else knows, but do you remember the color of the spores on the Cordonarius? I just showed you that one again next to the other one that I just showed. Does anybody remember that color?
They like rust colored.
It's like a rust color. It's this guy with those really rusty colored spores. Well, I'm hoping that I've got a spore print to show you, the moment of truth. And this is what I've got. And that is not a rust color. This is a piece of glass. Um, and if I hold the dart up behind it, maybe it's a little easier to see that what you've got here is a spore print that is basically kind of cream colored. It's got a little bit of a hue to it that's not quite white, but I do like using something that is glass or some aluminum foil or something that's really going to highlight what that color is because you'll find references to pinkish tan or pinkish buff or lightly pink. And sometimes it's really tough to tell the difference between those without having something that really shows that. And white paper or black paper is not always the best option. If that's all you've got, sure, make one with that. Or if you're trying to make an art project with that spore print, you can use that. But a piece of glass, a microscope slide is a really good option, or just a Pyrex bowl that happens to be the thing that you're going to forget you did a spore print in and then eat out of the next day. Um, this is Lepista nuda, the one that we're seeing here. This is a bluet. It's got other common names, but if you look that up in a field guide, you would see that that was Lepista nuda. So because we have this spore print, we could go into the field guide that I have right here that I hope you all own. Um, I'm not friends with them. I don't get a commission from them when I sell one of their books, but this is a really good group of people that have done lots of work on lots of groups. They're not just responsible for this generalized field guide for our region. They're also the ones that put together the specific guide for boletes that you see right here, which is also really helpful. These are not cheap books, by the way, when you go to buy this kind of thing. But that book is what, Christina, like 20 bucks or something? Um, Pretty inexpensive. But this is also the same group that worked on this milk cap mushroom guide that I have. Um, I think it's worth it for me because I'm teaching people about these things and I want to make sure that I identify these as closely as I can to what they are. But the reason I like this one is because it's got more species in it than our other regional guide that is also a good guide, by the way. I'm not an anti-fan of this other guide. I really like this one too. And there's a little bit more interesting biology stuff, I think, in the other one that you see here. If you need a reference for these, Christina's got them, I've got them, but either one of these or both of them would be worth investing in because they're so inexpensive. And they fit in a bag pretty easily. When I go out and try to carry all of these books, it's like I have a 50-pound weight on my back. So it's nice to have something that covers most of the things that you're going to see most of the time. I thought we might have time this evening to go through and try to key something out, but maybe we'll do that on Saturday with the walk. Even if it's with one of these things that I gathered yesterday, I think it's helpful to do that. I would love to do it with a milk cap or a bolete just because those keys are a little more extensive. The keys in this thing are not my favorite because they basically just get you to a section and then you have to just use the photographs within that section to narrow down what you have. So it will give you some good information about what the gill color is, or sorry, what the spore color is, what types of gills they have. It'll give you some good steering guidance, but it won't tell you down through a group of pairs to exactly what that species is. You have to make that call based on the photos. And of course, it doesn't include everything we have. I mean, we've got tens of thousands of species. You can't put together a field guide that would include all of that. So get a field guide for sure. Come out to these programs when you can. There's tons of good info online. Mushroom Expert is my favorite website. Michael is an English teacher and he's a very good writer and engaging writer. You'll read it and laugh, at least at something. It's not really sterile, it's not boring, but he's really particular about his ID. He doesn't tell you at all about whether something's toxic or edible, and that's because he's trying to get you an identification, not whether you're going to get sick or whether you can eat something.
Say the name
So,
again.
mushroomexpert.com, I think, is their
Expert.
website.
Thank you.
Really good site. He's up in the Midwest. He's not down here in the Southeast, but it's a terrific site. And I use all of these things, of course. It's not as if I knew that that was a bluet and called it a day. I always check, and I always check at least one reference and hopefully two or three if I can. Um, but just that spore color is enough to separate out something that's a choice edible from something that I know is going to make me sick. So that's important. But that's all I have for specimens this evening. I didn't want to put on slides and have anybody fall asleep on me. I wanted to try and keep you engaged a little bit. Um, it's a little new using the camera. I think maybe at some point I'll try to use the microscope and kind of prepare some sections of things next time. But it's kind of nice to see the whole mushroom as well. But again, questions are welcome today or Saturday, either one. And if
Brad,
you
Karen's curious if you've ever raised your own mushrooms from spores.
not from spores, and the only thing I did was, uh, well, I used to have kind of a grow chamber out at Caw where I would basically just huck a few chunks of wood in there that had some fruiting bodies on them and just see if I could continue to keep that mycelium growing. And it did work. Just threw a tarp over the top of it. That's not, of course, the thing you want to have in your garage or in your house. Um, the grow kits that you can buy. I did try that once with the Mushroom Mountain kit and didn't have a lot of success with that. I might have gotten some very small fruiting bodies that started, but you do have to be attentive with that. You've got to keep it at a particular temperature. You do have to water those things. They are effective, but you have to stay on top of it. And I'm a person who has at least 3 jobs and two 16-year-old girls and a wife and 30 bikes. So it's tough for me to stay on top of that kind of stuff sometimes. And honestly, if you go buy fungi from the grocery store, they are just as good and they're not that expensive. Um, these should be accents for meals. They shouldn't be the main course.
That's great. Um, we have a couple comments like Michael Patrick's excited to hang on Saturday and said, great job. Nora thanks you. And William Southgate just asked a question or a statement. There is an autoimmune disease in humans called scleroderma. Is there a relationship between the two?
Only in the roots of the word there, right? Like hardened skin or toughened skin. That's the translation, loose translation for that genus. And as you might remember, that was the one that had the kind of casing to it. It had kind of a tougher skin on the outside that enclosed those spores. So I'm sure that that is a result of that condition that you're talking about, maybe a symptom of that condition that you're talking about. I don't know it, but because it has that name, I'm sure it's got some kind of a manifestation that's similar to that description.
That's great. I was thinking that too, actually. So, Michael, I'm glad you asked that. Does anybody else want to unmute and ask Brad a question right now?
I was just going to say, I think there's a couple, at least one I know of, at least I knew of in Johns Island who raised mushrooms, a couple of local places that will raise edible mushrooms for you. They're often at the farmers markets and such.
Yeah, you just need some space, right? It's nice not to have to do that right next to where you live and sleep.
And humidity.
But that was what the monastery started doing at one point, right? They were doing oysters or something. I don't know if they're still in that or not.
Yeah, Judith Kramer, I think Judith was on here. She might know a little bit about Mepkin Abbey and they were doing some mushroom stuff for a bit. I'm not sure if she's still on here or not. Um,
I kind
but
of I kind of like the magic of like occasionally finding something out in the woods that I know is edible, that I know I've identified properly, and taking that home, and it just being kind of a special thing instead of, going to buy my batch every week or whatever. But it's not as if I don't like mushrooms. It's just not something I think to buy every time I go to the store.
I tried one of those logs once too, and I did not. I felt like it was like my bonsai when I was young that went wrong. I was like, I just didn't spray it enough. I didn't do it quite right. It just didn't work out, unfortunately.
Yeah,
But
you have to be focused. I mean, it's just like a garden. I mean, you can't just plant a bunch of stuff and not look at it again. You've got to tend to it.
yeah, agreed.
I forgot to share this. This was one of
Oh,
my
that's
one of my
fabulous.
girls did that, and she named them things like Sammy Stinkhorn and Patty Puffball. Um, that was a Father's Day gift, and I am leaving you with this because it's something that inspires me to know that I'm not a dictator when it comes to their education, but I like to see that I have had some influence on them and how they perceive that world. Um, these are not people too, but they certainly are deserving of our respect, and I'm happy to see that kind of application of that knowledge.
That's awesome, Brad. That is incredible. Thank you for sharing that. And thank you for your time and for sharing all this great information. There's a lot of comments coming in, like Craig saying the close-ups of the mushrooms were great. Terry said same thing. Camera worked out so well. Nice presentation. I think
Good.
everybody is super stoked. And this has been a really nice change from like the typical kind of PowerPoint. And I think we're all just love that you took the time to have, specimens for us. And then really it like worked out great to be able to see everything.
Yeah,
So thank
glad we
you
got out
for,
yesterday.
yeah, that's great. Wonderful. Nikki put the link in the chat for connecting y'all with Saturday. I will fortunately be out of town. I won't be able to be there, but I know you're going to be in great hands and the weather looks to be wonderful. 9 to 11 at Caw on Saturday. So I wish y'all well. And Brad, that's just awesome. Thank you for sharing and really appreciate all the wonderful information and love this format.
Of course, thank you all for being here.
All right, excellent. Well, everybody have a wonderful evening and have fun naturalizing, and we'll see y'all in the field soon.
Thank you.
No, no.
Thanks, y'all. Have a good
Thank
night.
you. Thanks, Brad.